Hello, my name is Anne-Katrin Mahlein. I'm from the Institute of Sugarbeet Research in Göttingen, and I'm a specialist in plant pathology. My group and I do research on optical properties of diseased plants. This is what my talk today will be about. I welcome you to today's PhenoRob Interdisciplinary lecture series on the impact of plant diseases on optical properties. So what will you learn in this lecture? We will talk about the basics, functionalities, and practical concepts of digital technologies in the field of plant disease detection. You will get insights into the impact of plant diseases on biochemical and biophysical plant properties, and how this impacts reflectance properties of plants. The focus of this talk will be on hyperspectral imaging, and as an important part, you will get an idea of how the data analysis approaches are implemented and how we interpret and link sensor information with biological processes. Since we do research for agronomy and crop production, the transfer of theoretical information to practical application in the field is also considered. We will close up the talk with identifying research gaps and open questions. Plant diseases are an important threat to crop production. According to a study of Erke et al., 12% of the potential yield of crops is lost due to diseases, 9.4% to weeds and 10% to pests. Methods of integrated plant protection are necessary to safeguard a significant amount of yield to ensure the production of food, feed, and fiber. Within this context, the detection of plant diseases and the diagnosis of the causal agent is crucial, also to decide for the right countermeasures. This task is often performed by human raters. However, with progress in digital technologies, this task can be automated by optical sensors and sophisticated data analysis approaches. But let's start with some basic principles. What we aim to detect is stress in a non-invasive and automated way. But what is stress? In physics, stress was introduced into the theory of elasticity as an amount of force for a given unit area. Since nearly 100 years, biologists have attempted to apply this terminology to biological systems. Within the context of crop plants, we differentiate among abiotic stress, such as water deficit, temperature stress, nutrient deficit, and many more, and biotic stress caused by plant diseases, insects, or mammals. In plant pathology, fungi, viruses, and bacteria are the most relevant and destructive pathogens, and in the following, we will focus on host-pathogen interactions of crop with fungal diseases. These diseases influence the crop in multiple ways. To perform and establish in a proper way, a crop plant has several elemental functions, shown on the left side. First of all, photosynthesis, transpiration, water and mineral uptake, and synthesis of metabolites and relevant compounds. When a plant is diseased, these elemental plant functions are impaired. You can see this on the right side. Typical symptoms such as shoot or leaf blight or leaf spots and wilt impair the photosynthetic apparatus and destroy plant tissue in a characteristic way. Root rot and vascular wilt impair the uptake of water or nutrients. This in sum causes stress and loss of productivity, a reduction of yield, or a total loss by the death of entire plants and areas in the field. To sum it up, the detailed impact of plant diseases are changes in the plant pigment, changes in the water content of the tissue, accumulation and degradation of metabolites like specific toxins, for example, changes in the cell and leaf structure, the accumulation of fungal biomass, and changes in the source and sink relation. And this in sum causes physiological and metabolomic changes and influences the optical signatures of plants. When we aim to detect plant diseases, what are the main tasks and steps? These are first of all the detection of plant diseases. The second relevant step is to identify a causal agent of a plant disease, then to quantify the amount and severity of the disease, and to monitor the occurrence and development of plant diseases during the growing season, for example. Normally or typically, this task is done by the human eye. So you have a look on a leaf and estimate the kind of the disease and the severity of a disease. If we now implement measurement technologies, which are imaging, but not with the human eye, we talk about the use of RGB technology, which uses the leaf reflectance in the visible range, or we talk about multispectral sensors and hyperspectral sensors, which also uses the leaf reflectance in the near-infrared range or in the shortwave infrared range. So let's have a deeper look on what happens on the tissue level of plants when light hits the tissue. And this is a follow-up of the PILS, the interdisciplinary lecture of Uwe Rascher. And to simplify the process, we have here shown the interaction of light with plant tissue by this look on a schematic sketch of a cross-section through a dicotyl leaf. If light enters the leaf tissue, it goes different ways. Some part of the light goes through the tissue. This is the transmission, shown in red. Some part of the light is absorbed, shown in green, due to the content of plant pigments. And what we are able to measure consists of the internal reflectance after diverse scattering processes and the direct reflectance from the leaf surface. If a plant is now diseased with plant diseases, we have several changes in the structure of the leaf tissue, but also in the biochemical properties. We have seen here in this example 3 different diseases. On the left side, you see the typical infection structures of powdery mildew, the Haustoria, the Aprestorium, Mycelium, and Conidia. And you see that the Haustoриum enters the epidermis cell layer. However, if you have a look on it, the cell structure is still intact. In the middle, you see the infection pattern of a leaf caused by leaf rusts. And you see also the infectious structures which are on the leaf surface, but we also have Haustoria and Mycelium within the leaf tissue, within the palisade parenchyma, and we see a typical oridinium rupturing the epidermis and the cuticula of the plant tissue. So here we have a slight destruction of the plant tissue. If you look on the right side, you see the typical interaction of a petrotrophic leaf spot diseases with leaf tissue, and here you see infectious structures. But these pathogens also cause toxins, and these toxins cause local cell death, and by this we have also chlorosis and necrosis in the tissue. And you can imagine now that if light enters now such a destroyed tissue, we have typical changes in the spectral reflectance pattern. And this is a process we make use of for identifying plant diseases based on spectral properties. However, if we aim to identify plant diseases of fungal structures by sensors, we also have to consider the scale. If you have a very close look to the structure, you will be able to identify single spores by, for example, hyperspectral microscopy. If you are on the leaf level, you will be able to identify characteristic symptoms. This is also possible on the plant level. On the canopy level, you have a higher distance to your object, and here it will be much more difficult due to the spatial resolution of the sensors to identify subtle symptoms which are in the size of millimeters. The same is true for the field. So here on the canopy and on the field level, it is more likely that you will find larger patterns of the diseases and the disease distribution over time. If we look from satellite data on the landscape scale, you will be able to identify large-scale patterns and large temporal patterns, but it will be hard to identify single symptoms or specific diseases on the scale. As mentioned before, the technology we talk today about is hyperspectral sensing. And if we make use of hyperspectral sensors, we have to consider the kind of the sensor technology behind. So we have either imaging sensors or non-imaging sensors. The technology can be applied in a proximal sensing way with a distance below 1 meter, or it can be used as remote sensing on a drone or on an airplane or a satellite. What we measure and what we cover by hyperspectral imaging is the visible range, the near-infrared range, and the short-wave infrared range. And within this area, we can identify and monitor biochemical variables, like the pigment content, for example, but also structural variables. And if a disease and the plant have an interaction, so if you have a specific host-pathogen interaction, these both parameters or factors are impaired in a very characteristic way with a specific temporal pattern. And this can be monitored and interpreted with respect to the disease development. But not only the scale is important, also the applied recording modes of the sensor systems needs to be considered. If we apply imaging sensors, we differentiate between whisk broom, push broom, or filter-based systems. And at the end, our aim is to obtain a hyperspectral imaging data cube with two spatial dimensions, y and x, and additionally for each pixel a spectral dimension z containing spectral reflectance patterns. So this is how imaging sensors work. However, there are also very interesting non-imaging sensors, and here we assess the integral of a specific area, so the mean reflectance of a specific area without a spatial dimension. Why are imaging sensors preferable compared to non-imaging sensors if we aim to assess very small and tiny symptoms of plant diseases? So using the spatial resolution of a sensor, we are able to differentiate healthy tissue from diseased tissue. This will be not possible if you only use non-imaging sensors, where you have the mixed information of healthy parts of your plant and diseased parts of the plants. In this scene you see here in the middle of the slide, we have a disease severity of 7.6%. So the rest of the tissue is healthy, and you can imagine that if we have a high amount of mixed pixels, the information you obtain is not as sensitive and as specific compared to imaging data based on hyperspectral sensors. Let's have a look on some spectral signatures of diseased leaf pixels. On the left side, you see a hyperspectral data cube of barley, and for each imaging pixel, we obtain a spectral reflectance curve, which you can see on the right side. Here, the range from 400 to 1000 nanometers is covered. In green, we have healthy parts of the leaves, and with the change in the color from green to light green to yellow to red, we have an increase in disease severity with a rust disease, and you see that this increase in disease severity changes the spectral reflectance pattern in different areas of the curve in a very characteristic way. So we have a decrease of reflectance in the visible range from 400 to 500 nanometers, a decrease in the range from 550 to 700 nanometers, and a steady increase from 700 to 1000 nanometers in the near infrared. But how specific is such a change in the spectral signatures if we compare different kinds of diseases? And to visualize that, I brought some examples from Sugarbeet, which are diseased with two different diseases here. On the left side, you see spectral signatures of Sugarbeet leaves inoculated with Cercospora beticula, causing Cercospora leaf spot. And on the right side, you see Sugarbeet leaves inoculated with Uromyces beta, the causal agent of Sugarbeet rust. And the spectral reflectance curves were measured each day, starting 3 days after inoculation until 21 days after inoculation. And we see that with ongoing time, we have a change in the spectral reflectance curve, which is very obvious for Cercospora leaf spot in the visible range. We have a decrease of the reflectance in the near infrared. And if we have a look in the shortwave infrared, we see that especially close to the water bands, we have an increase of reflectance with ongoing disease severity and disease progress of Cercospora leaf spot. If we compare that with Sugarbeet rust, we see that the changes are smaller and not so obvious. So we have nearly no changes in the visible range. However, with very high disease severities at later stages, we have a decrease in the near-infrared range for Sugarbeet rust at around 700 nanometers. And interestingly, in the shortwave infrared, we have a decrease of reflectance over the entire area. So it is not an increase as it was for Cercospora leaf spot, it is a decrease. And this is a very important information because we have characteristic spectrosignatures for different diseases, but this also varies over time. And these spectral reflectance patterns can be used for further data analysis. And here one example can be a clustering approach, which can be used for an automated data-driven analysis of hyperspectral images. And this was done here and is visualized on this slide. So we have our hyperspectral imaging cube with many pixels, and for each pixel spectral signatures. And here a simple k-means clustering was performed, and based on 6 different clusters, we were able to identify and differentiate healthy parts of a leaf from diseased parts, and even the disease spots have different specific regions which can be clustered automatically, and these regions can be also explained with biological meaning. So we have a center of a spot where sporulation took place, and we have the edge and the border of the spots, and this was identified automatically by this algorithm. We can make use of this information for several relevant research tasks or also for plant breeding. And for the case of Cercospora, we had an investigation of tolerant and resistant genotypes and used hyperspectral imaging to compare and characterize symptoms on tolerant or resistant genotypes. And here you see the results of that. We had 2 different genotypes. One genotype which was susceptible, where symptoms developed in a proper way, and this could also be identified by hyperspectral imaging. And then we had a second genotype which was tolerant, and here also symptoms developed. But when we had a closer look to the symptoms, it was very interesting that we had nearly no sporulation in the center of the symptoms, which is shown here in the genotype plus the QTL. So this was a very interesting hint that these genotypes have a lower sporulation, and a lower sporulation means that we have a lower disease pressure during the vegetation period, which is very positive for the epidemiological development of a plant disease. Since we now already know that we are able to differentiate plant diseases based on spectral signatures, and that we also get insights on the sporulation intensity of plant diseases, it will be also of interest to see if we are sensitive enough to also identify resistant plants against pathogen, because this is a very important task in plant breeding. And we had a study with barley and different genotypes which are susceptible, the wild type, or which produce a papilla as a resistance reaction, the MLO3 genotype, or which produce a hypersensitive reaction, the MLA1 genotype. And each of this resistance reaction causes different changes in the structure of the tissue. So the MLO3 forms a papilla which hinders the haustorium to penetrate the cell. The MLA1 genotype forms a hypersensitive reaction, and this is a necrotization of single cells, and by this the pathogen is not able to feed itself. So we investigated our tissue with hyperspectral imaging, we extracted the spectral signatures, and we developed spectral traces of the different genotypes. And it was very interesting to see that the susceptible genotypes had different spectral traces. This was due to the different disease development. But interestingly, the resistant genotype, which has the MLO3 resistance in the background, was very, very similar to each other. And this is because the resistant genotype causes no disease symptoms, and the tissue had a very, very similar phenotype, and we were able to measure this by hyperspectral imaging based on a fully data-driven approach. A little bit more challenging is a resistance screening of the hypersensitive reaction, because this is also a very, very tiny reaction on a cellular level. And here we also worked with hyperspectral imaging, and made images of the barley tissue on different time points, and we applied a data-driven approach on the data to identify the lesions which are nearly not visible by the human eye. So we started using our hyperspectral data cube, transforming the data in a matrix. We determined clusters with the highest distance to healthy, to the non-inoculated tissue, and estimated a simplex per cluster, which represents each cluster by few parameters. And with this approach, we were able to identify and localize at a very early time point the hypersensitive reactions seen here by these yellowish dots on the leaf, and by this it was possible to perform an automated phenotyping task of the host pathogen interaction. How can we transfer now this information and this knowledge into a practical application? So because the aim will be to go into the field and to sense plants and to identify plant diseases in the field. And that's a vision behind all our research. At the beginning, we have a picture with a specific sensor taken from a crop canopy. And on this picture, you see a wheat crop stand diseased with yellow rust. We use this picture in a machine learning approach or another data analysis approach to detect the disease on the leaf and to differentiate it from the healthy tissue. And based on this information, in combination with the location in the field, we can create a distribution map, and this distribution map can be used as an application plan. To test this theory, we performed a research on the field scale to do an in-field disease detection based on UAV data, and we investigated different diseases in wheat like yellow rust, brown rust, septoria leaf blotch, powdery mildew, or tan spot. And in this figure, you see that each of the different foliar diseases of wheat have very characteristic spectral signatures. In green, you see the spectral reflectance curve of healthy tissue. Compared to that, we have plotted the different diseases in red, brown rust, where we have a slight decrease in the reflectance in the visible range, an increase of reflectance in the visible range, and a steep change in the reflectance in the near infrared. And what is very positive is that also the other diseases like tan spot, yellow rust, or septoria have a very individual spectral reflectance curve and can be separated from each other based on their spectral reflectance pattern. However, that here is just a picture of one moment, so with one disease severity. If we want to draw the entire picture, we have to have a look on time series measurement, and we have to investigate how a pathogen develops over time. And therefore, time series measurements are of importance. And here you see time series measurements over a certain period of time. And I just highlighted for you the most relevant regions in the spectral signatures. And here in this region, we identified so-called time-independent turning points. So this is a turning point where we can identify that we have reached a very specific step in pathogenesis, so a very specific step in symptom development. And this turning point is very characteristic when the course of the spectral reflectance pattern goes a very specific way. And this can be the cause, for example, for brown rust, if we have the phase where the first chlorosis appears, and when this chlorosis erupture and the oridinospores appear, we can see that we have a time-independent turning point here from 500 to 700 nanometers due to a change in the color of the pathogen. And these time-independent turning points can be used to be applied in the field if you want to identify a disease in the field without knowing when the pathogen was inoculated. But all this is still very complex, and it is very complex to assess all this hyperspectral data, to store and to handle this hyperspectral data. So it is a very important question to ask whether it is important and necessary to measure with a hyperspectral sensor for practical use. So it could be an idea to assess in a scientific approach hyperspectral reflectance pattern to identify the most relevant wavebands and to reduce the complexity of the data to a multispectral data set with the most relevant wavebands. And this can be the initial information for the definition of sensors which are characteristic for individual host pathogen systems. And this approach was exemplary applied on the hyperspectral imaging data set of the different wheat diseases. You have seen some slides before. And here the approach was that after measuring the spectral reflectance curves, a feature selection algorithm was applied, and we identified that the number of 10 features is relevant to reach a high accuracy in the classification of the diseases in the field. So we need only 10 wavebands and not hundreds of wavebands, which is very important to reduce the data complexity. In the next step, we identified the most relevant wavebands and used these wavebands in a classification approach, and were able to create an application map with a lower complexity of the data. So this was the proof of concept that it is possible to reduce the data complexity from hyperspectral imaging to multispectral imaging. Another aspect to address when we go from the lab into the field is the high complexity that we can see in pictures which are from a canopy in the field. We have different environmental factors which impair our measurements. So the illumination intensity, for example, the light intensity may change due to shadowing and different illumination intensities. We may have wind in our crop stand, which can reduce the data quality. And you see here in this picture from above that we have different organs with a different distance to our sensor, and with very complex geometry. And that's very challenging if we want to analyze these data, because we have so many different structures. We have old leaves, we have young leaves, we have ears, we have diseased areas, we have senescent areas, we have shadowed areas. And therefore we need algorithms which are able to handle these high complexity. And some older algorithms like the spectral angle mappers are not really able to handle these complexities. So we need more powerful algorithms which are able to identify the heterogeneity, and we need also an interpretation of the heterogeneity. And therefore, you have to apply machine learning approaches like support vector machines, or even deep learning approaches with proper data sets and training data sets to consider the complexity and to have a correct classification of your data. So these were just some insights about the background of the interaction of plant diseases with the plant and how this influences the optical properties of plants. However, research is still ongoing, and we have a lot of remaining challenges and research priorities. And one of the questions which often comes at first is, how early could plant diseases be detected? And this question is difficult to answer because it depends on the setting of the sensor, on the size of the symptoms, and on the host pathogen interaction. But today we know that everything which can be seen by the human eye can be also visualized by a hyperspectral imaging sensor. But if these technologies are more sensitive or not is difficult to answer, because here we have the question how we develop training data and how do we ground truth the data. What is still ongoing is the consideration of crop geometry and the consideration of growth of crop stands and external factors, as shown on the last slide. And where we also need further research is research on the interaction of different kinds of stressors. So how do multiple stressors and multi-disease infections influence the optical properties of the plants? What is also not known so far is how invasive plant diseases influence optical properties of plants. So what we really need are spectral libraries as a collection of different and characteristic spectral signatures of different diseases, which can be then used to identify an unknown setting. If we now have the detection technologies running, and if we use these in the field for example, site-specific application, it is still unclear how a spot application as a disease control influences the epidemiology of the diseases, and what this means for future registration of pesticides, for example. At the end, I also want to mention that we need a communication to the end users. So we need training for farmers regarding all these new technologies. And it's still an open question if the farmers should use these systems by themselves, or if these kind of systems can be provided as a service through crop managers or through advisory services. Yeah, with this, I'm at the end of my talk today. It was just a very brief wrap-up about optical properties of plant diseases and how it can be measured by sensors. I thank you for your interest, and I'm looking forward for your questions.